Okay, let's start, everybody. Welcome to this morning session of the Bio Summit Saturday, the Bio Art and Design track, which will continue in the American afternoon after the plenary session. But this is a pre-plenary session we planned because there were so many people sharing great projects that they wanted to present. So we have room for 6 talks here today, but maybe we'll start with a short introduction of myself and Paige, who are the moderators. Paige, do you want to start with introducing yourself shortly?
Yes, hi, I'm Paige. I'm French American. I'm currently based in Riga. I was part of the Bio Fellows cohort this year, and I'm involved in helping organize all this stuff. I'm really excited about this panel. I'm a super biodesign nerd with no background in science, but trying to interview all of the really cool people that are doing it through a podcast that I'm just starting. And that's me. Go ahead, Roland.
Cool. Yeah. So, and I'm Roland. I'm from the Netherlands. Just moved to Norway, and I'm interested in art science collaborations for myself, and also in facilitating these in others. And I've done so before by teaching the Biohack Academy at Waag in Amsterdam, and by organizing other workshops and things. Thanks for the snaps, Georg. But for now, we will start shortly with presentations to give you more time. Every person that will present, which will be, I can go to the next slide as well. This is the technical stuff. I will go through this as well. If you just entered, change your screen name to Jane Doe, she, her, so your pronouns, your affiliation, and your country. It doesn't have to be Jane Doe, but really add your own name. These are the websites. You all know this from yesterday. This is the thanks to the sponsors of the event. And now we're at our session. Start your day with Bio Art and Design. Yes. So these will be our 6 speakers and our moderators, which we just introduced. So the speakers will be in the following order, not on the screen. We'll start with Shihan Zhang of the Alter Studio, then Damien Boue of Nemeton Biolab, then Aliki Kostopoulou. I hope I pronounce your name somewhat right. Please also pronounce your own name better. Of Conscious Gems. Then Kwan of MIT. Paloma Marquez of the National University of Tres de Febrero. And we end with Revathi Ravishankar, a bioartist from India. And so everybody has 10 minutes and then we have to go on to the next one. So I don't know if there's a lot of room for questions, but you can of course send your questions in Zoom, but you can also continue this conversation later on this in our Bio Art and Design Slack channel and keep it going. And also quite a few of these artists show works at the Bio Creation Station online, so you can also see their exhibits later online. So that being said, we're going to start with the first speaker. And I'll do the slide, so you just have to say next one and I'll click.
Oh, thank you, Roland. So morning from San Francisco. I'm really glad to be here virtually with you all to share my project and the ideas. And thank you for the BioSummit community inviting me, and also big shout out to Roland and Paige, and also the whole team who's supporting us along the way and make this happen. Next slide, please. Next slide.
All right, it's my role.
Yeah, so my name is Shihan Zhang. I am a design futurist based in San Francisco, California. And my passion lies in challenging cultural stereotypes and the social preconceptions through the building experience of future-present intersections in order to address Anthropocene challenges. Next. I co-founded Auto Art Studio, where we speculate possible futures to address unseen challenges and opportunities. Next. Now I am a member at New Inc., an art incubator lead by the New Museum and New York City, became my upcoming projects. Next. My work mainly focusing on emerging environmental challenges, such as global warming, air pollution, new materials, as well as emerging technologies, such as bioengineering, artificial intelligence, and mixed realities. Next. I believe that design is a method and a medium to inspire people to look forward positively, as well as thinking critically. I position my work as a bridge that allows audiences to access the unsurprising challenges in a more curated experience. Next. And today I'm going to talk an emerging unsurprising challenge that we have, the data storage. Next, please. According to a report from IBM, 90% of the world data had been created in the previous 2 years, and the data growth continued to explode. It is estimated that each person will generate 146 gigabytes per day this year. Once you're aware of how all the data you created as a single individual, you can start to imagine just how much data we collectively generate every single day. Next, please. And by 2040, storing digital data is set to create 14% of the world carbon emissions. Next. So how can we find a more sustainable way to store our data? Next. Imagine that you can store data of your entire life on your skin. Next, please. It might sound crazy, but the skin consists of skin cells and each cell has DNA, which is the key to make this idea possible. Currently, there's an emerging technology called DNA digital data storage that code data by base pair ATCG from a DNA sequence, and let's see how it works. So firstly, the coding part, it translates binary code to biolanguage using ATCG. So you can see the A equal to 00, G equal to 01, C equal to 10, and T equal to 11. Next. And then we build DNA strands matching the sequence using a DNA synthesizing machine. That's how we store data on the DNA. Next. If we want to read it back, we can decode the DNA strand. Basically, it needs to run through a sequencer, which returns generic code and then translate back to binary code that can be read by computers or other electronic devices. And here's a diagram shows comparison between the hard disk, flash memory, and the DNA. You can see the DNA storage has strong advantages, especially for long-term stability and competitive capacity. A few kilograms of DNA could theoretically store all the data on this world. Next. We figured out how to store data on the DNA, but how we add to your skin. It can be simple as applying cream, just like your skincare. Next. Technically, it needs DNA editing technology, and the CRISPR is the most popular one, which commonly used to alter DNA sequence and modify gene function. There has been several successful experiments. For example, a team at Harvard University has used the CRISPR to encode video into E. coli. Although there has not been any test on other living things beyond the bacteria, it's not hard to imagine that it can be possible to other animals or even us. Next, thanks. Therefore, we made this project called Skin Database to explore how humans could evolve our body to handle the continuing data explosion. Next, please. We imagine that due to the data overload by the year of 2040, hardware-based storage are no longer able to support the data we keep creating. Therefore, a machine called TAC has been invented and become a normalized solution for people to store their personal data. Next, please. Just like how you store documents into different folders on your hard drive, you can store data on multiple areas on your body, depending how private this data are. Next. In order to load the data onto your body, firstly, you need to transplant a genetic engineered synthetic skin tissue called memory tag. There can be multiple memory tag function like a folder in order to respond to different data storage. Then you will need to purchase a tactile machine, a device that functions as a converter and bioprinter that transfer binary codes to DNA strands and produce genetic modified chemicals such as your skincare that you can apply to your skin. Tubes in the center actually contain necessary materials for synthesizing DNA strands. Several plugins extend from the device, allowing user to pair with some like the memory tags that they transplanted on their body. You may wonder, why would we buy this, or why we won't store data on your skin? It actually has many advantages. Wherever you go, your data is always with you. Second, and it's easy to store or retrieve from the skin compared to internal organs or other tissues. Third, the interaction is user-friendly. It's just like you apply your skincare. And then lastly, it's not inherited, so you don't worry about your children carrying your DNA if you don't want them to. However, there are also some side effects. You may at risk of health problems of DNA mutation, or your safety will be concerned if you are carrying confidential data. Last but not least, be careful with your intimate relationship. Next. All of this information is included in the super long manual. It contains the mechanisms, step-by-step instructions, as well as the details and the sequences you need to be aware of. Next. We are collaborated with a film director on a video piece showcasing the experience of Ashley. So a tech user who uploads her memory onto her body as a routine. There's video, maybe unmute Roland.
I think it's best for the time if you share the video in the chat and people can see it afterwards.
Yeah, sure. Yeah, I can share it in the chat.
Thank you so much, Shihan. That was a beautiful presentation. I love to think about skin, like just your skin as a canvas, and I love the applications of it in biodesign. And you wonderfully mapped the challenges and opportunities of doing something like that. So now we're moving on to Damien Boue, a fellow Frenchie. Welcome. He's going to be introducing to us the concept of tree shaping and talking to us about their experimental garden near Grenoble, I guess, and also talking about a software about how others might be able to do this. So take us away.
Hello everybody, it's nice to see you all, and thank to all the team who's organized this BioSummit. I'm Damien Boue from France. I'm actually at the city of Grenoble. I'm the coordinator of a STEM non-profit organization named Nemeton. We are specialized in biology, biotechnology, and the environment. We are based in the city of Grenoble, and our main purpose is to open a biolab, which will open in next January. Next. One of our collective projects is an experimental garden whose name is the Jardin de Lougue. In that place, we do have experiments with a technique whose name is tree shaping. This technique allows us to grow trees directly in the shape of stool in this first case. The trees are actually Paulownia, a variety of tree which grow really fast. I think it's the probably the tree who grow faster in the European climate. Next. We guide those trees during their growth along a set of tutors. The proximity between the trees will cause multiple contact transplants. when the shape will complete and the wood thick enough, we will cut out the stools and remove all the excedent branches. The resulting stools will ready to use, and their production have consumed more CO2 than they have produced. Next. The first generation will probably crop next year. This is why I do not have, unfortunately, any picture of them for now. This winter, the Nemeton community will work on a new software whose goal is to generate little 3D printed junctions to link the tutors and create way more easily new shape to grow. It will allow designers to make new shape to grow way more easily. They don't have to make one by one each of one of the pieces will link all the tutors. We can see on this slide a software close to what we would like to create. It's actually a plugin for SketchUps whose name TruthFab. It is developed by the Asso Atner Institute, who is based in Netherlands, I think. And we will maybe work with them. It's not sure, but it's definitely this kind of software we would like to create. Well, I was way more fast than expected, but that's it. We grow stools and table and stuff like that.
Super cool.
Thank you, Damien.
Any
Maybe
questions?
because we have a bit more time, we can open up. Maybe first get what you presented. Is we can also have some more time for Q&A until we have to go to the next speaker, which is nice. Actually, I might have a question. So this TruthFab, which we now see, which is this plugin for SketchUp. Maybe can you just slowly explain one more time, kind of, because I use SketchUp quite a lot. We use it for the Biohack Academy as well to build our tools. But what does this plugin we see here allow us to do, and how does this relate to your tree project? Like how? Okay, just because here we see a table made from bottles, right?
Yeah. The idea in our development, we will change this kind of software and replace the actual bottles we can see there by some conventional tutors, a string of metal, very cheap tutors, this kind of tutors.
Cool. Maybe it would be nice if a link for some online thing like a website to share in the chat afterwards, so people can see more of this project, because I think it's not on the Bio Creation Station, but I think it's an amazing project. And with that, I think we have to go back to the next, or along to the next speaker, but Christiana said then we will write on time. So thank you a lot again.
Thank you to you.
Great, and our next speaker is Aliki with her project Conscious Gems, creating a grid of regenerative homes of consciousness. And there we go. Please explain.
Hello, everybody. Good morning or good afternoon, depending where you are. So to start with, I would like to challenge a little bit your scientific thinking, infusing a little bit of spirituality and a little bit of permaculture into today's presentation. Next, please. So Conscious Gems is practically a project that I have been creating, or it has been creating through me, let's say it better. And it applies to different systems that are also used in nature. And I will start from the system of regeneration from the macro to the micro, or the micro to the macro. This project has come as a guideline, as a blueprint. And it starts with the pyramid of Maslow that most of you most probably know. And this pyramid has to do about our needs. And then this pyramid can be translated also with the 5 elements, starting with the earth, and each element is connecting to one specific action. So the earth is connected to permaculture, for example. Earth, metal, water, wood, and fire create a whole circle. So automatically we're going from the traditional pyramidic systems that we used to have in our organizations into the circular ones, because the one element is actually holding and creating the next one. And that's how it's happening in nature. And if we translate it to our projects, we can start having it in our project too. So the whole idea is, how do we translate nature into what we are doing, how we are functioning? So we don't have systems growing and dying and growing and dying, but we have whole circles of regeneration. So we create, we rise, we fail, and we become mycelium to feed back other projects and give our information to new cycles of us. So the pyramid to the circle, we can see that the individual system, the small triangle, is becoming part of a circle. That can be a group, that can be your organization, that can even grow further and become a node of a bigger system that is working in the whole globe. And many systems together are creating a whole ecosystem. Exactly the same as the nature is doing it. A single cell is a full organism, but all together with other cells will end up creating an organism, an individual, a person. That part of a group is becoming a bigger system, our team, our company, and then this all of a sudden is becoming part of our ecosystem, of the country, of the nation, of the world. Next, please. So this was the first layer, from the macro to the micro and the micro to the macro, to see ourselves as part of the ecosystems. And the second one that I would like to introduce are the cycles. We all function by cycles. And actually this system came to be by pure observation. Like 2 years ago, I went and I watched my calendar, my entries in my calendar, and I started realizing that each month, for some reason, had a pattern. So in some months, I was like super outgoing, meeting people, having meetings, spreading the information, everything. In other months, I was just observing or working in myself, like deep work, deep focus. And then I realized that was repeating every year. So then I looked deeper. And then I started realizing that these patterns were somehow connecting with the zodiac signs. And why is it? Because the zodiac signs, they're just a translation of the constellations that are happening around the planet in one specific month. It has nothing to do with characteristics and everything that we have attached it to. It's practically how our planet is moving in the solar system. Next one, please. So starting to overlay cycles, I was curious to see if there were other relationships that we couldn't see or we didn't know or I didn't know, and how I could apply that to my life and to my projects. So I started looking at layers and layers of cycles that are working in the earth and see if there are correlations. And in this one that you are seeing, the macro, is you're seeing the bigger picture with all the layers, and then we will start zooming in slowly. You can see the principles of permaculture, which is 12 principles. And I was like, let's see if any of those might be connected to any of the other characteristics that I could see. And then I started saying, and I would think that some of the principles, for example, the use and restore energy, have the same characteristic of the zodiac sign that is representing this month. Next one, please. Going deeper into the cycle, I was realizing that the seasons, the 4 seasons, have specific characteristics, right? In the winter, we are more inside ourselves, towards where we are going right now. when in spring we want to go out, we want to expand. And in summer, we are in full blossom, in full openness, going into the autumn, that we are starting slowly coming closer to ourselves and harvesting what we have created. And deeper into the cycles, please, you can continue to the next one. I was realizing that our projects themselves are having patterns that apply also to our full one-day cycle of our bodies from dawn to dawn. So our project starts with the first light and with new ideas, going into creating something, putting more energy into it. That is the early morning, which is the time, according to Ayurveda, that we have the more experience to be outgoing and actually talk about our projects. And then it's going to the middle day, which is our metabolism is the highest possible. And that is the time that we can create new teams, expand and communicate, going towards the sunset, that we start retrieving inside ourselves and we want to relax and get closer. Do you remember autumn? That's the moment we are right now. It's the sunset of the period of time, right? And they are going into the night that we just want to chill, share, and be in a comfortable space. Remember winter? Same thing. And then we're moving into recuperation that is happening, the incubation during winter, to go into a new day again. Please, next slide. So all of that apply both to the plants, to ourselves, to nature, to everything that is around us. And I will just go a little bit faster. I have shared that with you anyhow if you want. You can see 12 months. You can see one day, you can see the moon cycles with 28 days, you can see the week with 7 days, and you can see one day. According to the organism we're looking at, we have to apply different types of cycles. If it's a smaller organism, a person, a small project, you can apply the daily cycles or even the moon cycles. If it's a bigger organism, an organization, a nation, a very big project, you might want to apply multiple year cycles. But still, there are layers over layers that are functioning at the same time. Next slide, please. We can continue the next one. This is the moon cycle, so we can talk a little bit more. This is the weekly cycles. Next one, please. And I would like to just look a little bit how this has been applied until now into the project, because I was looking back actually, and I was realizing that without even wanting to, the nature itself was creating it, and it was applied anyhow. So it started last August with the seed, practically seeding the first idea of the coherence research. Going into incubation last January, it started creating something inside a space, right? And that was in Davos in Switzerland. Third step in April was the flowering. It was creating the blueprint, the team creation, going outwards, bursting the first flower. In June was the time to practically go into obtaining the yield. And that was the first time we moved into a community in the south of West France. And we started really interacting between us. And then we're going to September and we're going into harvesting whatever has happened and understanding how many people are on board and which are the gifts of them. And we're moving forward and continuing applying all these principles. We're going into a new incubation, but this time there's a second layer because the seeds are different. The seeds are more than the first seed last August. And continuing that, we will grow, we'll continue growing naturally and according to nature. And this is practically a proposal for all of us to understand that the right action at the right time will eventually create a cycle of birth, expansion, death, and rejuvenation without even us having to put any more effort. So it's practically an invitation to see when and how to act so we can be in a flow with the universe instead of contrary to it. We can be together with it. So thank you very much for that. I hope it's inspiring for all of you. And if you have questions, please let me know.
Thank you, Aliki. That was wonderful. I love to think about biodesign and permaculture and how they work together. And it's really great to be reminded of that. Next, we're moving on to Kwan, who works with weeds and what they evoke, from what I understand. So
yeah, I have to stop my screen sharing and do it again because Kwan has a movie, and I have to check that I share my audio. So I have to do share computer audio, share computer sounds, and then screen 2, share computer sound, and now it should work. So let's test
Great,
this.
thank you. Yeah. So good morning, everyone from Boston. I'm from Boston. My name is, I'm based in Boston. My name is Queenie. I'm from Hong Kong, and I'm currently doing my graduate studies at the Art, Culture, Technology program in the Department of Architecture at MIT. Today, I will just present a video work. So I hope to bring you all a very slow and meditative morning through the journey of weeds. We can try the video now. Yeah, great.
In the continuous and necessary debates of geopolitical conflicts, Anthropocene and climate changes, plants have been constantly thrust into the spotlight. Examples are those of the botanical gardens, migrating seas, rare species born out of extremity, and the strategy of urban greening as a state manifestation of power. Plants witness humankind colonial savage. They inspire us with their queer embodiment, providing historic traces of human global expansion. Sometimes one may even see hope in plants. I have been tracing weeds in different parts of the world, starting from Hong Kong, my home, from which I move outwards to unfamiliar lands, communities speaking languages that I'm foreign to. Weeds outline the territory of marginality in our landscape. Sometimes I feel like a visual anthropologist, following an ethnographic investigation into a possibly primitive group of plants. Perhaps it's similar to how anthropology develops its own beauty from science, supposedly through a more humanistic and descriptive approach. Weed is the daily greenery that can be found everywhere. But why would a weed be defined as a weed, not a plant? The notion of weeds rejects the conventional biological classification into genera and species, but a collective group which squeezes through cracks in concrete surfaces and drains. Weeds signify autonomous solidarity and nomadic vitality, evading dominance and control. Shooting weeds is an exhausting process. As you can see, they all look familiar, trivial. But in fact, diving into the details, their nuances are beyond imagination. Because the visible is mundane, the act becomes the crux. When something is flattened onto a 2D image, the eyes behind the lens need to consider the temperature, the texture, and the timing of the shooting. Every image is a microscopic encapsulation of a macroscopic social mechanism, a kind of quantum sociology, a resemblage of quantum physics. It demands humbleness, withdrawing one's self-consciousness to greet this grandest form of survival in the most miniature format. Ultimately, every result is unique and full of randomness. I could read into them for hours, and search for the creed. But what is the creed of the weeds? In the beginning, it was just about their vitality, their charm, their unflinching strength that grew through constraints. Yet in endless repetition, beyond the physical embodiment of shooting, I land on unsettlement. There is a bottom-up, discursive, almost accidental effort to bypass the sovereignty of order, to stroll along the periphery of dominant structure, to drift within dismantled discourses, returning to the agency of matter. How can a noun migrate to a pseudonym? How can we withdraw the term weeding from the dictionary? How can we play the language game of Wittgenstein? How can a phenomenal observation be translated into a new identity? Weeds are often gleaned at the boundary, so one could see it both inwards and outwards. Unorganized and guerrilla, they speak of a certain degree of complicity and emotional detachment. I am always fascinated by just looking at weeds. They are performative, vibrant, dynamic, whispering many untold stories, those of intricacy and contingency of our times. The weeds tell the possibility of performance to exist as an everyday practice, performance as reenactment. A spirit of non-compliance, transgressing boundaries, inhabiting limits and cracks, never shy from thriving regardless of triviality, moving between circles, difficult to divine, impossible to get hold of, formless. So I think about the relationship between manhole covers, paving slabs, construction barricades to weeds. I think about the temporary labor contracts, false long hours, and the inexorable rights to safe distance, to the rights to be masked. I think about the proletariats in us and in nature. I think about weeds again. To imagine a system of faith beyond imposed politics, is this just a myth of alternative identity? What is the creed of weeds, beneath which is a stamp of political absurdity, a post-colonial casualty? Independence is dismissed as a threat to national security, while a submission to the power is to be despised. Instead of being a second-class existence in a foreign land, I have chosen the weedest state. They stand still at the edge of time, in plain sight. Their silent composure is contested against the cacophony of history. In alternative worlds that they etch, we carefully approach through an outdoor choreography that weaves fermented perspective, that dares you to bow in front of a dichotomy between nature and culture, to be transported into an unknown dream freely and unconditionally. When ontology is gravitated towards text but mediated through image, perhaps we could think through things as much as working through things. The latter stands as a temptation. The former is yet a necessity of revival. In every shivering moment I return to Wheeze, an ongoing that conjures up a suspension in our daily spectacle, propping into frozen frames, long exposure, single and multiple temporalities. Looking at Wheeze through looping, zooming, scaling, pacing, and expediting is, to me, a rhetoric of contemporary resurrection.
Wow. There's so much to rebound on here, but that was just such a beautiful poem. Thank you. Weeds are such an underrated species in general, and it's so wonderful that you gave them front stage here. Paloma, it's your turn now. Paloma will be speaking to us about the collaboration between minerals, artists, and technology.
Hello to everyone. Thank you for being here. I am an artist and a researcher at the University 13 de Febrero in Buenos Aires, Argentina. I'm so happy to share with you my investigation where I create artworks using minerals and technology. I call this art category lithotech art. Even though minerals and rocks aren't live organisms, they are essential for the natural systems. Next slide, please. I start this investigation taking pictures of the rocks that were nearby, analyzing their weight, their colors, and their texture. But then I could study their physical properties as their conductivity and magnetism. That takes us to the next slide. Here, I wanted to show you some links that are surround the rocks, as the times that are very different from the human times, the lithological cycles, the territories that I will speak about that later, the mineral vitalities. It was fun that some of the other art projects talked about similar things than mine. The analogies and relationships between men and minerals, that points make us think about the unbalanced relationship between humans and minerals. And the contradictions that are inside the rocks, for example, the stillness and the dynamics. These topics are some useful things that I use to develop my artworks. Next slide, please. As I mentioned, the actual relationship between human and minerals in this anthropocentric era is not an horizontal and empathic as we would like to. This thought led me to think of the possibility of a lithocentric point of view, where the minerals are the main actants. None of these centric point of view should be, instead of any centrism, we should look for an horizontal relationship between the agents. Next slide, please. The lithotech art is an art category that I present here. I think of this project as an horizontal collaboration between minerals, technology, and artists. In this case, we can see this image that shows us in one side, the primitive mineral, and in the other one, the mineral after the process, like the electronic circuits. Next slide, please. I wanted to show you some of the artworks that I collect and I think that are related with my investigation. For example, this artwork is called Songsmith and it was created by Jenna Burchell. She took a fragmented rock and reconstructed with gold. When your hands get closer to the rock, a unique composition can be heard, recorded in the rock original place. Next slide, please. This is another artwork that it was made by Marcela Armas and Gilberto Esparza. They call it Sideral. They created a technological device that senses the magnetic field in the surface of the meteorite. The data obtained displayed an audio they created, inspired by the sound of the place where the meteorite impacted. Next slide, please. Please press play so we can listen a little bit. Thanks. This is an artwork that I made a few years ago that I call it choreographic erosion, where a mechanical arm moves the rocks over another one, making an artificial erosion. Meanwhile, the vibrations are sensed by a piezoelectric crystal and they are amplified. This installation is composed by 3 devices. I call it choreographic erosion because I think of them like a dance or a dialogue between the 3 devices. I am also interested of the sound as a mineral identity sound. Next slide, please. And if you can, please let's play. This is another artwork that I call Osho's collaboration. In this case, the mineral is part of the electronic circuit. It is an essential part of the oscillators. It is composed by oscillators and amplifiers. Its conductivity produces an unpredictable sound that depends on the position and the properties of each mineral. In this case, the minerals and the technology works together, and also me as an artist. And I like to think that this artwork is getting closer to an horizontal relationship because I give the mineral an autonomy to make any sound. This can be used as a music instrument or also as a performance device. You can watch these videos and the description of these artworks in the video creation station. Next slide, please. And to finish, I wanted to show you the future of this investigation. That's what I'm looking at, what I am searching right now. The possible territories of the minerals. I think that the rock is a territory itself of movement and actions. Each rock carries its own landscape in its surface, and it also constructs a landscape in its way. So we have a diversity and possibility in this theme. I am studying the scales specifically, which are very interesting for me. One is the microscale as a texture and its atomic activities, for example, the radioactivity decay. And the other one are the macroscales that take us from the landscape to the cosmic territories. All the territories are related with the lithological cycles and the attraction and repulsion forces. I like this graphic because it shows the symmetry and also the opposite opposite of the scales. Next slide, please. Well, this research is part of my thesis of the degree in the master degree in electronic arts at the Universidad Nacional 3 de Febrero. And if any of you are interested in this theme or are working in something similar, please don't hesitate to tell me, because I am very interested in connecting you. And thank you for the team that facilitated this meeting.
Thank you, Paloma. At a geological scale, it feels like minerals are almost a living thing as well. It's just really cool to think about collaborating with that.
Yes, I am carefully thinking about life. It's more vital matter, as Jane Bennett speaks in their book. Yes.
Love to talk more about that with you. Revathi, it's your turn. You have a playful session for us. I think you wanted everyone to take out some paper and pens. Here's to you. Finish us off.
Hi, a big good afternoon over everyone present there. Well, it is good evening in India. I'm Ravishankar. I'm an upcoming bioartist in India. I have transformed the structure of microorganisms with unique designs and patterns. So in the upcoming slides, you'll be able to watch this, and I think you will enjoy it. Next slide, please. So this is the first slide here is this the typical E. coli. These two images that I have drawn by my own hand, which I have not changed the original structure, but I have implemented new designs and patterns so that you can view this organism in a unique style. As you all know, there's nothing but E. coli is a gram-negative organism, but when you see the organism as a design or pattern, it will be a totally different area for you. So you will not you will view this organism as a pattern and design, not as a typical microbe which we used to study in our lab or in college or whatever. Next slide, please. Next slide, please.
What is this? Sorry.
No problem. We'll be right back.
Shall I proceed?
Sorry, I have a Google problem. I'm sharing my second screen again. It's some kind of pop-up. Present. Then I have to move this there again. And we're here. Next one. We're here.
So, I have the most respect and love towards the bacteriophage. I don't know why, but whenever I see this organism or study about it, I really feel getting excited. Just see the structure, the center one, which is the typical bacteriophage that we all see in we study in our textbooks, the web pages. But I didn't change the original structure. Here I've used unique designs so that we can see this organism in a very different style. Next slide, please. So Noctiluca scintillans, this is a bioluminescent algae. Whenever its original cycle gets disturbed, it emits bioluminescence. It is seen all over the world, especially in estuaries. So it will be a beautiful one it will be a visualized treat whenever you watch the bioluminescent algae. So it emits blue color. So the center one is the original structure. The other two slides on the left and the right one is the one which I have drawn by my hand with designs and some styles. So one more important factor about Noctiluca scintillans is that it can, with the help of this algae, you can definitely estimate that B body level is really high in that particular area. So whenever you see a bioluminescent algae, that means the oxygen level is very low in that water. So this is used as one of the indicator in estuaries especially. Next slide, please. So coronavirus, the hot topic at present in the world. a single organism has made down the whole planet to get shut down. So the coronavirus is one of the most powerful virus, as we all know that. So here I have used a special pattern called mandala pattern. Just it will be a right. We can use the circles. With that, we will develop a unique set. It will be uniform throughout. So these are two different varieties. Base is the mandala pattern, by which I have made the coronavirus structure. You can also visualize it in Google, but I have not made a very different styles, but I have made a very simple one so that it will be easy for us to visualize that. Next slide, please. So I have made a bio out of various fungi. The first one, that is this one, is about Aspergillus fumigatus. The second one is about Trichoderma viridae, and the third one is Penicillium chrysogenum. The last one we all know that which produces antibiotic penicillin. So here also I have used some different styles and structures. So the base structure is not changed. I have used some patterns so that you can view it as an bio art. Next slide, please. So influenza virus, this is also one of the most powerful virus. For this also I have used the mandala pattern, but with some different styles. So when you compare it with the original, I have made some slight modifications. The original structure is not changed, but I have made some slight modifications, so it will be a visual treat to watch. Next slide, please. So Paramecium caudatum, as you all know, it's a parasite. Here also I have used some unique patterns and designs. So designs, it's a treat to watch this organism under microscope. But when you draw this, I feel really extremely happy, and I have made some made some designs with which we can differentiate the organism. Next slide, please. So here comes, please be ready with your paper and pen. So here I'm going to ask the questions. I'll give 30 seconds. Please try to give the answer. Okay, the first question. Who was the first person to crystallize Tobacco Mosaic Virus? Anyone ready with the answer? I'll give 30 seconds time.
Can we Google it?
Anyone ready with the answer?
Google
it. It was Sir Vandal. He was awarded a Nobel Prize in the year 1945. He was shared the Nobel Prize. He is the first person to crystallize Tobacco Mosaic Virus. So shall we proceed with the next question? Which organism produces the antibiotic Cephalosporin? Any idea? Anyone with the answer?
I think it's a fungi.
Yeah, it's from fungi. It is derived from the fungi Acrimonium, originally which was called Cephalosporin. Acrimonium chrysogenum. So third question. This is a very interesting topic. When you study about this in Google, you will get drenched in this organism. Scrub typhus is caused by which organism? Scrub typhus is a fever which is caused by the larval bite. It is caused by a bacteria, namely, this name itself is a very funny name. Orintia sushugamushi. Orintia sushugamushi. It's the organism which causes this fever. This bacteria gets infested or manifested in the larval bite. So when the larval comes and bites the human beings, the bacteria gets ingested in the human being. It's very dangerous. Once you get this fever, if you are not able to identify within 3 days, it will become very fatal. The fourth question. Which organism causes lymphatic filariasis? Any idea? Which organism causes lymphatic filariasis? So the answer is Uchredium bancrafti, a parasite. So the fifth question. What are the special features of the Gonococci and Meningococci? It's nothing but these two cocci are gram-negative cocci. Usually you heard the name Cryptococcus, Cephalosporin. They are all gram-positive, but these two cocci are gram-negative. So coming to the sixth question. Which staining technique is used to detect TB? So since it's a microbiology tuberculosis, you all know that it possesses high amount of lipid molecule. So use a special technique called acid fast staining. That's nothing but Ziehl-Neelsen technique, with which you can able to stain Microbiome tuberculosis and identify that. So next question. Cryptococcus neoformans belong to which group of organism? It's nothing but a fungi. This also causes a very fatal disease, namely the cryptococcal meningitis. And coming to the 8th question, what is the selective media used for the identification of Cephalococcus species? It's nothing but mannitol salt agar. This is a special media which is used for the isolation of this Cephalococcus species. It produces golden yellow colonies on this media. So if you have MSA with that, we can easily isolate Staphylococcus aureus. So 9th question, which organism is responsible for monkey malaria? So this was a currently hot topic prevailing all over the world. It is caused by Plasmodium noelsi. So that is the parasite which is responsible for monkey malaria. Next slide, please. So I have just given it as underlined words. You have to identify the organism. Since it is very 30-second left with me, I will let out the answer. The first question, that is organism Pseudomonas aeruginosa. Second one is Orintia sishugamushi. Third one is Xanthomonas campestris. Fourth one is Yersinia pestis. Fifth one is Leuconostoc mesenteroids. Sixth one is Noctiluca scintillans. Seventh one is Geotrichum candidum. Eighth one is Streptomyces griseus. And ninth one is Trichoderma viridae. Here it's,
Thank you, Revathi.
Thank you so much for providing me this opportunity so that I met all of you and able to share my bio art with you all. Thank you so much. Any
Wonderful.
feedback
I
is
hope you
welcome.
have drawings of all these things that you made us learn about. This is the end of the session, and we've got to take just a group shot of everyone. So let's see. Heidi, are you ready to take a screenshot? You said you were able to do it. If you want to like pose on the count of 3, that would be great. Turn on your videos and just like wave to the camera and make a face.
So there's a few of us, so I'm gonna just hold on. I'm gonna make my screen bigger, and I'm gonna change to the other side. So when I say 1, 2, 3, ready now. Next one.
Maybe we can do one more. Can we do one with a group hug? That I learned recently. So you all have to do kind of this, and then it looks really cool. So you hug the camera, right?
So who takes a picture if you're all hugging? I'll take the picture. Don't worry.
That's more like I'm a bear or like impersonating some kind of scary animal.
Yeah.
We got it.
Oh.
Thank you so much for your presentations. Those were really, really cool projects.
Yeah, thanks everyone. So let's continue the conversations on the Slack. And there's so much more to say. And it was really inspiring to watch all the diversity of projects. And now maybe let's all take a break because the plenary is starting in like 25 minutes. So it's nice maybe to take a little walk and get some air. And then, yeah.